Welcome to the microscopic world of glutamatergic neurons within the hippocampus. The exciting world is infused with electrical and chemical activity at the synapse. We will illustrate today the communication between two pyramidal neurons. The synapse, consisting of a presynaptic terminal, a postsynaptic density, an astrocyte, and the synaptic cleft, are within a tortuous environment. The presynaptic membrane of the CA3 neuron is full of vesicles, approximately 200, containing the chemical neurotransmitter glutamate. The postsynaptic density, PSD, of the CA1 neuron usually has over 100 to 200 channel receptors upon its surface. Today, we will only show a few for visual acuity. The action begins with the arrival of a speedy 0.2 meters per second action potential, the brain's electrical current, at the presynaptic membrane. Watch out! Once the membrane has been depolarized by the action potential, the voltage-gated calcium channels open up and the extracellular calcium enters the cytosol. The influx of calcium launches a biochemical cascade that induces vesicles to dock with the presynaptic membrane. Vesicles that are already docked, approximately 10, fuse 2 to 5 per second with the presynaptic membrane. Each vesicle fusion causes a release of approximately 1,200 glutamate molecules into the synaptic cleft. The volume of glutamate being released into the cleft equals 2,500 to 6,000 per second. As glutamate diffuses throughout the synaptic cleft, it binds to glutamate receptors, including AMPA, NMDA, and K-NAT receptors within 50 microseconds. Glutamate is cleared within 80 microseconds out of the synaptic cleft. When glutamate binds to two homeric or heteromeric subunits of the AMPA receptor, the dimer upon dimer ligand binding clamshell closes 21 degrees, thus partially opening the channel. Now that the channel is open, extracellular sodium flows through the channel and potassium leaves the intracellular space. Binding of glutamate to each additional bound subunit induces additional opening of the AMPA receptor channel, and thus causing further depolarization of the postsynaptic membrane. When the agonist glutamate or NMDA bind to the NMDA receptor subunit NR2A, the clamshell-shaped ligand binding domain of S1 and S2 closes 24 degrees. The closure initiates torque upon the receptor, and it conformationally flips open the channel to a partially open position. Then D-serine binds to the NR1 subunit coagonist or glycine site, and the clamshell-shaped protein dimers of S1 and S2 close 24 degrees. Once all of the subunits are occupied by their coagonists, torque will induce the NMDA channel to fully open. Even with the channel fully open, cations cannot flow through the NMDA channel because of a voltage-dependent block by magnesium. When the postsynaptic membrane depolarizes to -50 millivolts, the affinity of magnesium for its binding site decreases, causing the magnesium ion to fly out of the channel. Watch out! Finally, the NMDA channel is fully open to calcium-sodium influx and potassium exiting the intercellular space. The calcium influx can trigger biochemical cascades that change gene expression patterns needed for synaptic plasticity, excitotoxicity, learning, and memory. Thank you for coming and learning about the exciting microscopic world of glutamatergic neurons.